Race it to the top.
You're already up there.
Exactly. Come on, Milo. You okay?
Yeah, I just can't jump that high.
You'll get it sooner or later.
I've been getting it for weeks. Can I ask you something?
Sure.
Do you ever think it's weird that we're from the same litter, but we're built completely different?
I guess. I never really thought about it.
I have.
You want to know why?
Yeah.
Then let's check someone who actually knows. What seems to be the concern?
I can't jump like he can, or run as fast. My legs are shorter. I just want to know why.
That's a fair question. Milo, you have what's known as the Munchkin mutation.
Mutation? That sounds serious.
It's not dangerous, it just means that there was a small change in your DNA before you were born.
So this isn't random?
No, it comes from your genetic code or the instructions that tell your body how to grow.
So something in my instructions is different?
Exactly.
More specifically, it is a change in your UGDH gene, which controls a protein called UDP glucose 6-dehydrogenase, which is responsible for synthesizing glycosaminoglycans. These are critical for your bone growth. The first step in this mutation is the first step in protein synthesis, or transcription. This takes place in the nucleus, as this is where the DNA is, the code for genes such as UGDH. When making a new protein, the enzyme helicase is first used to unwind the DNA, allowing an mRNA to come through and copy the template strand. Once it's finished, the mRNA will be an exact copy of the coding strand, but replaces the thymine bases with uracil bases. To create this mRNA molecule, the mRNA strand utilizes RNA polymerase to create the matching base pairs with the template strand. But instead of bonding adenine with thymine, it bonds adenine with uracil. For example, if our template strand had bases AAG, CTT, ATG, and GGG, our mRNA strand would have bases UUC, GAA, UAC, and CCC. This is the exact replica of our coding strand, replacing the T bases with U bases. This results in our mutated gene. Now in your case, Milo, your UGDH gene goes through the mutation of having a deletion of 3,300 bases and an insertion of 216 bases. This results in your UGDH being mutated. Now, this may be too much to think of in terms of numbers, but if we go back to our mRNA example, we could apply these same mutations to it. For example, if our guanine base in the second codon was deleted and in that place an insertion of 3 bases happened, such as AUU, then our strand would become mutated, resulting in the UGDH gene changing.
Okay, but how does a change in instructions actually change something like my leg length?
To understand that, you have to look at how those instructions are copied before they're used.
After the mRNA strand is finished coding, it makes its way to the ribosome, which is made up of RNA. Once the mRNA goes inside the ribosome reads the mRNA in codons, or every 3 bases. Its job is to pair up tRNA outside the ribosome that contain the anticodons of the mRNA codons. For example, CCC pairs with GGG, and UAC pairs with AUG. The reason these tRNA strands are important is because they carry protein chains that create amino acids with the codon, and these amino acids can then bond with each other with polypeptide bonds to form proteins. If we take our example mRNA strand, we can predict the type of amino acids that it will create. In this case, it creates phenylalanine, glutamic acid, tyrosine, and proline. If we take our mutated RNA that we applied our insertion and deletion to, we can see that it creates the amino acids phenylalanine, isoleucine, tyrosine, and proline, switching glutamic acid with isoleucine compared to our original mRNA strand. These changes can ultimately affect enzyme stability and enzymic activity. In the case of UGDH, this can result in the shorter growth of leg bones in cats and is now called the Munchkin cat mutation. Another way you can think about this is if a composer wants to share his music piece with the entire world. If he wants to, he might make a copy of his original work. You can think of this as transcription. He could then take this copied sheets and send it to production facilities that put the finishing touches on it to spread it to the world, mimicking translation. However, if there is a mistake made when copying the original sheet, this can result in the production facility being slowed down or completely stopped, resulting in the music piece not being released to the people. This would be representative of a mutation.
So a small change in the genetic code changed how my bones developed?
Yes, even a tiny difference at the molecular level can affect how a trait appears.
So we didn't do anything wrong.
There's no wrong here, it's just changed.
Is it going to cause problems?
You may not jump as high, but you're healthy. You just move differently.
He's actually faster underneath objects than I am.
That's because I don't have to duck.
See? Different structure, different advantages.
I guess I just wanted to know the reason behind it. Do you think people notice my mutation?
Probably.
And what would they think about it?
They might like it.
Excuse me, is that a little munchkin kitten?
Yes, he is.
I've been looking for one. They're brothers?
Indeed.
I'll take both.
Wait, both?
Told you.
You can't separate family.
Guess we're going together.
Obviously.